Swayam Prabha. Digital India. Educated India.
Welcome to my course on genome editing and engineering. Today we are going to discuss about TALEN, which is a editing technology, little bit different from the last technology that we discussed, the zinc finger nucleases. The word TALEN is also a hybrid between TAL effectors and the word nuclease. In this lecture we are going to discuss about the basics of this technology platform. So, we will start with a discussion with TAL effectors, their biology, and how these TAL effectors are finally fused with nuclease proteins to produce artificially made talents. So, let us start with one interesting molecule which has been listed as molecule of the month at the time of recording this lecture. So, every month protein data bank highlights a protein molecule or a complex as a molecule of the month. So, this month the non-homologous adjoining supercomplexes has been listed by PDB as such. You know about the DNA double strand break mechanisms and how the DNA double strand breaks are repaired by the cellular mechanisms. Amongst them the NSEJ is one of the major players, and this molecule is also complex is also important from the point of view of the TALEN technology, because once we use TALEN to induce defined double strand breaks on DNA sequences, it has to undergo repair either by the homology repair pathway or the NSEJ pathway. Similarly, a few years back in 2014, PDB listed these TAL effectors in December 2014 as the molecule of the month. Today we are going to spend our time in discussing at large about these TAL effectors. So, what are these TAL effectors? So, TAL stands for transcription activator like effectors, and these are produced by several types of bacteria who inject these proteins into plant cells. The tails reach the plant cell nucleus and activate genes that make the host susceptible to infection. So, they are infectious entities. However, during the course of evolution, as a response to infection by bacteria and the repertory of tails they use in the pathogenic mechanism, plant cells have evolved a way to resist this attack, and when such plants are injected with TAL effectors, they immediately activate special resistant genes that protect the plant. We will not be discussing about this defense mechanism, because our interest is just restricted to the use of TAL effectors in the development of the TALEN technology platform. But we will discuss slowly about the tail biology and how they are involved in the pathogenicity. These TAL effectors bind to double stranded DNA through a central domain of tandem repeats. So, we are going to repeat these tandem repeats again and again, and there will be a lot of redundancies in this discussion, but they are important from the conceptual point of view. So, transcription activator like effectors specifically bind to double stranded DNA through a central domain of tandem repeats. We have to remember the central domain of tandem repeats, which is very, very important for understanding the function of tail effectors. Each of these tail effector repeats comprises around on an average 34 amino acids, and they recognize one specific DNA base through a highly variable residue at a fixed position in the repeat. The amino acid sequences of the repeats are nearly identical. Besides amino acid position 12 and 13, these are called as the repeat variable diresidues. So, out of these 34 except 12 and 13, everything else is identical. Sequence analysis and structural studies have revealed the molecular basis of DNA recognition by the tail repeats. So, repeats with different RVDs with different amino acids at the 12 and 13 positions. So, different DNA base pair preferences, and consecutive RVDs in a tail correspond directly to the DNA sequence in the binding site, resulting in a simple one repeat to one base pair code. The basic building block of tail protein we have to remember is a helical hairpin. Tail proteins contain a series of alpha helical hairpins arranged in a continuity or tandem. Each of these alpha helical hairpin domain of tail proteins binds an individual DNA base, as we have already discussed. Overall, a single tail protein would recognize a very long specific DNA sequence. On this right, you can see a space filling drawing of a TAL effector bound to double helical DNA, which is colored in orange, and the DNA reading domains of the TAL effector is being shown in blue, which is wrapped around DNA. Let us learn a little bit about the discovery of tail. Who are the people who discovered tail? In 2010, two groups who were working independently and in plant pathology, one led by Bogdanov at the Iowa State University, and the other by Jens Bock at the Martin Luther University in Germany, found out the role of tail in causing infection in plants by pathogenic bacteria Xanthomonas, and they also deciphered the mechanism of how these TAL effectors recognizes host DNA. The list of TAL effectors are quite large, and you can see here some of them listed in column 1, and each TAL effector has a particular target gene to which it would go and bind, and they are part of a target gene family. And if you are interested to know about the details of each effector, you can go to the papers published by the original authors, and the full references are available in this publication by Deb et al. Let us just focus on one or two of these. For example, you have a effector called TAL-B and another called TAL-C. TAL-C, the target gene is OS suite 14, one of the very, very famous genes. Then you have others like PTH-XO, so and this also binds to OS suite 11, and then others like AVR XA7, AVR XA23, and they target various members of different gene families. So, these particular bacteria, which is an infectious agent, enters the plant leaf through the stomata or any mechanical wounds caused by maybe insect injury or other mechanical injury, and then sprayed within the crop by mechanical contact or by rain and irrigation water. And when it finds a favorable warm wet condition, the organism will cause rapid and severe disease in the plant. And these are some of the symptoms you can see in some of the host species. For example, in philodendron, you can see the bacterial leaf blight caused by Genome campestris, and you can see the clear symptom over here. And this is the cereal crop rice, in which the bacterial blight has happened due to infection by Genome oryzae, Patovar oryzae. This organism is very important in the understanding of the tail effectors. In general, Genome includes a large group of plant pathogenic gram-negative bacteria, which infect more than 200 different plant species, and we will show you only just two of them in the earlier slide. And they rely on various effectors secreted through different types of protein secretion systems. In particular, the type 3 secretion system, T3SS, to suppress host immunity and obtain nutrients from the plants. In oryza, Genome oryzae, the T3SS effectors are grouped into two types: transcription activator like effectors and non-tails, which are also called Genome outer proteins or JOPs. So, we know that tails are virulence factors of the plant pathogen Genome, and these are translocated via a type 3 secretion system inside the plant cell, and once inside the plant cell, these tails are subsequently imported into the nucleus, where they bind to specific DNA sequences and transcriptionally activate gene expression. And most of the times these causes susceptibility in the host, but sometimes the plant as a response to this type of infections will produce some kind of a defense system, and these very tails may in fact bind to the immune response genes and help the plant in fighting off the infection. We are not going to discuss on that part much in this particular lecture. Let us have a little bit of detailed view of the rice, Genome oryzae, Patovar oryzae or Jew interaction with focus on effectors during pathogenesis. The effector proteins secreted via the type 3 secretion system, this is the type 2 secretion system. We are not going to study about it. We are going to study about only the type 3 secretion system. So, the effectors, T3 effectors, which are secreted via the type 3 secretion system, you can see these T3 effectors, and these are actually pumped inside the plant cell through these beautiful apparatus. So, these are directly delivered into the plant cell cytoplasm, and these effectors consist of TALEN effectors, which are DNA binding proteins that upregulate plant genes, leading to further susceptibility. You can see over here, and then classical example is sweet gene upregulation in Jew rice interaction. Another class of T3S effectors, the non-TAL effectors, are involved in dampening the immune response by targeting defense signaling pathways, working directly or indirectly by binding to plant proteins. So, this is the pathway by which the immune response is triggered. The plant would counter these effectors using multiple mechanisms, and it involves the executor R genes, whose transcription is activated by TAL effectors, leading to strong immune response and resistance. and by resistance proteins that target effectors directly or indirectly. So, let us learn a little bit about the bacterial protein secretion system. We already know that there are at least two such systems, the T2SS and the T3SS. Bacteria deploy a multiple array of secretion systems, and we can divide them into different classes based on their structure, function and specificity. So, some of these systems are conserved in all classes of bacteria which secrete a broad array of substrates, while others are only found in a small number of bacterial species and are specific to only one or a few proteins. So, you can see here so many different types, type 1, 2, 3, 4, 5, 6 and 7, and all of them are different, and some of them can be grouped into larger groups. So, in general, protein secretion from a bacteria is a two-step method. So, here sac or tet is involved in the first transportation step, and we call it the sac or tet dependent mechanism. So, here the proteins will cross the inner membrane with the help of either the sac or tet secretion pathways, and then they will land up here in this periplasm, and from here they are transported across the outer membrane using a second secretion system. Okay. So, this is a two-step sac or tet dependent mechanism of protein transport. Many gram negative protein secretion system transport their substrate across both bacterial membranes in a one-step sac or tet independent process. Sac or tet is a two-step transportation system, but some of the bacteria do not use that particular system. It uses a one-step sac or tet independent process, and such systems are okay. The pathways of the secretion system T1, T3, T4 and T6 contain the periplasm spanning channels and secrete proteins from the cytoplasm outside the cell. However, the mechanisms are quite different. Three of these, the T3, the T4 and the T6 can also transport protein across an additional host membrane. So, this membrane belongs to the host, and these three transportation systems can inject through the host cell wall or membrane and deliver the secreted proteins directly into the cytosol of the target cell. So, let us see a little bit in detail about the various secretion system of Xanthomonas and their functions. So, the T2 system is responsible for host cell wall degradation. T1 is responsible for adhesion to host cell and extracellular matrix. T6 is involved with the microbial competition, and T5 adhesion to host cell and auto aggregation and biofilm production, and then the bacterial competition, bacterial conjugation is carried out by the T4SS. Amongst all these, the T3SS is involved with the host cell manipulation and phytopathogenesis, and this is the focus of our study. So, you can see here all these systems have diverse components and mechanisms. We are focusing our discussion on the T3 secretion system, and the Xanthomonas T3 secretion system contains a HRP, hypersensitive response and pathogenicity gene, that encodes T3 secretion system. The HRP type 3 secretion system is essential for pathogenicity of most Xanthomonads except xylem restricted Xanthomonas albinilians. So, This T3 secretion system spans both bacterial membranes and is extended by a hollow conduit, the HRP pilus, that traverses the plant cell wall. Bacterial effector proteins are secreted through these T3 secretion system and translocated into the plant cell cytoplasm via bacterial translocation complex that inserts into the plant plasma membrane. Interference of type 3 effector proteins with plant cellular pathways happens. So, in A you can see the type 3 effectors elicit and suppress plant defense responses. Plant pathogenic bacteria translocate effector proteins via the T3 secretion system into the plant cells through the pilus. In resistant plants, individual effectors are directly or indirectly recognized by corresponding plant R proteins or activate the plant R genes, and which elicits a defense response, and these are designated as the ETI. The indirect recognition of effector proteins by plant R proteins depends on effector mediated modifications of the plant target proteins, which are sensed by matching R proteins. Several type 3 effectors suppress the ETI and or PTI responses and thus promote bacterial virulence. PTI responses are triggered upon recognition of bacterial pumps by plant PRRs and do not only interfere with pathogen survival, but can also affect T3 secretion mediated delivery of the effector proteins. This is the overview on known plant targets of type 3 effectors. The translocated effector proteins interfere with the assembly of cytoskeleton, then MAPK cascades, and then gene expression, proteasome dependent protein degradation or hormone signaling pathway. So, these particular T3S system is a conduit for so many different functions happening inside the host plant. Let us now briefly look into the role the TAL effectors play in the bacterial infection, which was first identified in Genome and Ralstonia. We already know that these TAL effectors are produced by pathogenic bacteria and they infect over around 200 different plant species. These TAL effector genes are ubiquitous in members of the genus Genome and Ralstonia and have apparent critical functions in a number of diseases. And the tail proteins are injected into plant cell which enter the nucleus via alpha or beta importance. There they play roles as transcription factors by activating expression of target genes to promote susceptibility or suppressed effector triggered immunity ETI of plants to facilitate bacterial infection. The TAL effectors bind to DNA specific site by DNA binding domain, and with the activator domain it regulates gene expression. Mesovirulent tails from Ju activates the expression of multiple rice sweet genes to promote susceptibility, and TAL7 from Jock activates the expression of OS09 Z2900 to suppress the AVR-XA7 induced resistance in rice. So, we know that the TAL effectors bind to DNA specific site by a DNA binding domain, and with the activator domain it regulates gene expression. Both Ju and Jock harbor large tail repertories, usually exceeding 8 in Ju isolates and over 20 in and around 20 in Jock isolates. The number of the tails present in a particular Genome species or strain varies from none to several. The transcription activator like effectors in Genome AVR BS3 PTHA are the largest effector family in Genome species and was identified by Robert Stahl and co-workers in 1990 at the University of Florida. The AVR BS3 was the first isolated member of the type 3 effector proteins in Genome species. The term TAL effector was introduced much later in 2006 to describe the large family of type 3 effector genes related to aviar BS3 from what was known at the time as Xanthomonas campestris, Petaver vesicatoria. So, with this we come to the end of our part A. Thank you. In the next part, we will be discussing about the various domains in the TALE effector proteins.